Hello, everyone. It's our great pleasure to welcome you to today's session, Overcoming the Challenges of Sample Collection in the Time of COVID-19. Thank you for joining us. While participating in the webinar, please keep in mind that you will be muted, but can submit questions at any time through the chat box. Our speakers will address your questions during the question and answer period at the end of the talk. This presentation will be recorded, and all attendees will be sent a copy following the webinar. I'm Elizabeth Firth, Human Genomics Product Manager at DNA Genotek, and I'd like to introduce today's speakers, Dr. Rafal Iwasiow and Mr. Scott Rabuka from DNA Genotek, and Dr. Bernadette Wildemore, who is joining us from P23 Labs. I've had the pleasure of working with Dr. Iwasiow and Mr. Rabuka for more than a decade, and we're pleased to have the opportunity to share our experience in how safe at-home sample collection is aiding in the work against SARS-CoV-2. I more recently met Dr. Wildemore, and I'm thrilled to have her join us to share P23 Labs' experience and success in validating saliva for their COVID-19 test. Today's agenda will explore our solutions across the SARS-CoV-2 testing spectrum, research efforts at DNA Genotek supporting COVID activities, a real-world case study featuring validation efforts conducted by P23 Labs, with time at the end to answer some questions. Dr. Iwasiow joined DNA Genotek in 2006, and since then he has held positions across research and product development, where he contributed significantly to the company's patent portfolio in numerous areas, including nucleic acid stabilization methods and physical sample collection devices. Dr. Iwasiow advanced his career through director-level roles within the organization, resulting in the appointment to his current role of Vice President of Innovation and Technology. Under his leadership, the research and development team contributed to successful 510K clearances of Oragene-Dx, the first and only non-invasive DNA collection device cleared by the FDA for at-home direct-to-consumer use. Additionally, he and his team have developed new chemistries, products, and service offerings throughout his career with the company. Prior to joining DNA Genotek, Dr. Iwasiow completed a postdoctoral fellowship at the National Research Council of Canada in the areas of neurogenesis, neuroprotection, and he holds a PhD degree in cellular and molecular medicine from the University of Ottawa. Mr. Rabuka is the Director of Genomics Program at DNA Genotek. He joined the company in 2008 as the product manager for the Oragene product and successfully expanded the product into a complete family of solutions for global research and clinical use. He was responsible for the launch of the company's first FDA 510K cleared product in 2011, as well as multiple new products designed to address evolving global market opportunities. In 2014, he assumed the role of Senior Sales Manager at DNA Genotek, where he established a global customer base for the company's genomic services and microbiome offerings. Mr. Rabuka earned his bachelor's degree in applied mathematics with honors from the University of Waterloo in 1997. Dr. Wildemore joined P23 Labs in 2020 as their Chief Medical Officer, after working alongside P23 Labs founder in several previous iterations. She has long been passionate about molecular medicine and microbiology, with a special interest in women's health projects. Dr. Wildemore completed a postdoctoral fellowship at the combined Harvard Brigham and Women's Hospitals program in molecular genetic pathology, and she holds her MD degree from Thomas Jefferson University Hospital in pathology, as well as her master's degree in microbiology. She is genuinely excited to be once again working alongside her colleague and friend at P23 Labs in the fight against the novel coronavirus. Without further ado, I'd like to turn it over to our speakers. Mr. Rabuka.
Thank you, Elizabeth, for the great introduction and for giving an overview of the talk today. I'm really honored to be here talking with all of you, and also alongside Dr. Iwasiow and Dr. Wildemore. I want to take a few minutes to talk about who we are as a company. DNA Genotek is a subsidiary of OraSure Technologies. We were acquired in 2011. Prior to that, we probably started commercial operations about 10 years prior to that. I've been currently with the company for 12 years, and it's been an honor to work through the portfolio growth and market development we've seen happen over that span. So OraSure Technologies has two main business units. The one on the left we're showing here is a Molecular Solutions division. Within Molecular Solutions, we have arms focusing on human genetics and also on microbiome sample collection. Within the last year and a half, we've also added a company from Belgium, Novosanis. They have a device for collecting first void urine. And then within our microbiome program, we've added some of the industry-leading microbiome service providers in Diversigen and also CoreBiome. So our focus within the molecular products side has traditionally been providing high-quality DNA from saliva samples. So a donor or a patient can spit, and from that saliva you'll get very good DNA for all kinds of downstream molecular testing applications. We use that experience within sample collection from saliva to look at other sample types and other analytes as well, and that is what has been spurring on the growth within our portfolio. On the infectious disease side, we have platforms for rapid point-of-care diagnosis of HIV and HCV. That's within the OraQuick platform. That platform has also been used for Ebola and Zika applications. So as you can see, I'll talk about this in a moment. We have a broad spectrum of looking at molecular testing applications, going right through to antibody and antigen applications. So this is really important. As a company, I think it's important to articulate our commitment and what we want to be remembered for, I guess, in many years to come. So we have proven expertise in bringing innovative solutions to address the world's most critical health challenges. From sample collection through testing, we are working to help in the fight against COVID-19. And that's really the focus of today's talk, is to talk about how we're able to respond to this pandemic and the needs that we're seeing from the market right now. So within our portfolio, we have a great array of products that can be used for the needs of the market right now. So on the left side, we have sample collection. So within the DNA Genotek portfolio of products, there's products already on the market that are very well suited and have been validated through a few EUAs now for COVID testing. So it's the OraCollect RNA, OmniGene Oral, and Origene DX product. I'll talk about these in a bit more detail shortly. And then on the infectious disease side, those platforms can be used for antibody and antigen testing. They're currently under development, and I'll speak about them in a few minutes either as well. The commonality here across both portfolios of collection kits is on many fronts. So, we've always focused on very easy to collect samples. We've focused on samples that are well protected and stabilized. And in regards to the COVID pandemic, there's other aspects you need to look at in terms of protecting the sample, protecting the individuals that will work with the sample, and ultimately easing the burden on the healthcare system. So this is a nice view in terms of explaining how we see some of the testing or market needs within COVID-19 virus. So what we're showing here is just the stages of infectivity of an individual and different analytes or biomarkers you can look at as the individual or patient would be infected with the virus, and then how you would potentially detect whether they had it after or not. So, viral RNA is present in an individual for, in some cases, up to 21 days. Following that, then the antigen, the antibody, can be available for testing as well. So within again, our portfolio of solutions for PCR testing, those products, OraCollect, OmniGene, and Origene DX, are available. And then under development, we have solutions for antigen and antibody testing to detect whether a patient has been infected in the past with SARS-CoV-2 virus. So just to focus on molecular solutions for the viral testing, I wanted to talk specifically about a few products that I've mentioned already. So here, we talked about the easy to use sample, non-invasive, high quality. We've always spent a lot of time understanding, the full path of the sample takes from, acquisition and recruitment, if you're thinking about research studies, through to, getting a sample from a remote location to a lab. Once it's in the lab, to, avoid cold chain, to be able to, be compatible with several different instrumentation available within the lab. So, those are some core attributes of products that we bring to market. And then, if you just look quickly at the instruction for use here, the OraCollect product is a sponge connected to a double-ended cap. Within the tube, there's 1 ml of stabilizing reagent. So the collection protocol is to rub the sponge on either side of the mouth 10 times, and then invert the sponge into the tube, shake it, and then it's stable and ready to be sent to the lab. With regards to the Omnigene Oral and Origene DX products, they're both saliva-based products. Depending on the product, the patient will spit either 1 ml or 2 ml of saliva. Within the funnel lid, there's a stabilizing reagent, so after the patient spits the sample, they press the lid down, the chemistry releases into the tube, and again stabilizes the RNA or DNA in the sample for the time that it takes from the collection location to the lab, whether that's at home or in a remote clinic or in a hospital for that matter. So the Omnigene Oral 505 product, a couple of specifics around it. We talked about the protocol for it. In the past, we've seen it used for other viral collections, so we have a list of viruses that our customers have used it for, and again, more recently, there's some good validation work on the SARS-CoV-2 virus. The OM 505 product is a 1 ml saliva kit product. There's an integrated barcode into it, and you'll see the tube on the right is the tube that is received in the sample. So again, it's very lab friendly or automation friendly. There's several robots that have been optimized or shown compatibility with this tube, and likewise several extraction reagents have been shown to be compatible with this chemistry as well. The OraCollect RNA product. Here again, you can see a closer up view of the chemistry within the tube. Similar, once the sponge is inverted, it's a tube that's very compatible with different automation solutions in the lab, and just like OM 505, the product's been validated for many downstream applications, and also for the SARS-CoV-2 virus. The chemistries used in OraCollect RNA are similar to what's used in OM 505, so we'll see validations where they're almost used interchangeably. So I've talked a little bit about some of the key market challenges from sample collection right through to result. We've always really liked this quote, and I wanted to share it again here. This is from Carolyn Compton, who was at the time with the NCI as the biorepository chief. She says, "We now have the technical ability to get the wrong answers with unprecedented speed. If we put the wrong stuff into the front end of our analytical pipeline, we will not only lose the war on cancer, we'll pollute the scientific literature with incorrect data that will take us a long time to sort out. This is a crisis that requires disruptive innovation." Our point in all of this is that there's no need for a lab to receive a sample that has been degraded or is of poor quality or wasn't handled well enough from collection to the lab. Everything that we do and focus on is to make sure that the sample is the highest quality from the point of collection to the point of running the analysis through the workflow in the lab. Just a final slide before I pass this over to Dr. Iwasiow. He'll be speaking about this a little bit more as well, but when we think about workflows, getting into the lab, there's some products and solutions that we worked on to be able to bypass extraction. So this is a product called PrepIt Q2A, which stands for Quick to Assay. It involves a series of heating and dilution steps within the labs. It's very quick, 30 minutes start to finish, and you can avoid extraction. So, particularly right now, there's been a shortage of some extraction reagents. So, in the event that you can bypass that, to satisfy that restriction, but also to improve your workflow and time from processing, arriving in the lab, and to the result. It's a nice solution for that. So a number of customers have been validating this, and Rafal will walk through this protocol in a bit more detail. So with that, I'd like to introduce you to Dr. Iwasiow and pass it over to him. Thank you.
Thank you, Scott. Hello, everyone. I'm pleased to share with you our R&D efforts in support of COVID-19 efforts. So leveraging our many years of experience in collection and stabilization, we have been supporting our customers interested in pursuing testing for SARS-CoV-2. DNA Genotek has been focusing on the technical performance of our portfolio of products, while our customers have been doing a lot of the biology and assay development. I will share with you 4 areas of research that are supporting the current COVID pandemic. First, I'll share with you R&D data in support of inactivation of SARS-CoV-2 using our consumer-safe reagents that are in our collection devices. We will then move on to work we have been doing to support extraction and processing of samples using traditional extraction kits, as well as novel reagents that DNA Genotek has developed to allow for bypass of extraction, which Scott just mentioned. And finally, I'll describe to you some of the work we have done over the years to support home self-collection. So first, let's jump into inactivation of SARS-CoV-2. We have done some extensive testing using cultured infective SARS-CoV-2 virus under BSL-3 conditions. We conducted an endpoint dilution assay for all of our stabilization chemistries, and we are pleased to report that the R&D results demonstrate that the DNA Genotek chemistries can inactivate the virus upon collection, providing an extra layer of safety to the patient, shipping, and laboratory personnel. Additionally, it should be noted that we achieve this without using any harsh reagents such as guanidine. Our reagents are consumer safe. As you can see from the bar graphs on the left, the dark bar represents the positive control. This in comparison to the negative controls that are throughout on the right, the hashed bars. And in comparison to positive and negative, you can see that the chemistry spiked with virus at the same levels as the positive controls, exhibit no sign of cytotoxicity, and they are at the same level as the negative virus controls. This work has been done in triplicate, and you don't see any error bars because there were simply no cytotoxicity to report at those levels. Based on the endpoint assay, we can easily say that the chemistries can inactivate the virus greater than 99%. So the next question we get a lot from our customers is really, I have your devices, I'm doing the collection, how should I process those samples? So one of the technical things we've been doing is to answer this is evaluating many different extraction kits. And, the answer that we can provide is that our chemistries are compatible with most of those extraction kits. We have tested a number of commercially available kits, as these days people are using all various different extractions, and we have found that many are directly compatible. Here's just a representative summary of some of the kits we have tested. We make no claims regarding the performance of these extraction kits with SARS-CoV-2. but we do test to ensure we get good nucleic acid recovery and no carryover of inhibitors. We have observed that not all kits, even from the same manufacturers, have the same level of compatibility or performance. This illustrates the need for proper testing, and DNA Genotek, through our technical support team, is capable of supporting our customers on this topic. I encourage anyone starting these kind of studies to reach out to our technical support hotline, and we can discuss the specifics of the kit you're using. I'd like to touch on the topic of the alternate option to extraction, PREPID Q2A, as Scott mentioned. It is a two chemistry component, liquid component, very simple to process. So on the right, you will see the workflow. So imagine here you get an OM505 saliva sample in the lab, standard procedure for all our samples, and an extra layer of precaution to the lab is a 56-degree incubation step for 2 hours. Once that's done, a small 100 microliter sample can be transferred into a deep well. At that point, should be heated at 75 degrees for 10 minutes. And the two reagents, 10 microliters of one and 20 microliters of another reagent, are added, vortexed, and left to incubate at room temperature for 15 minutes. The sample will separate at this point, and the top layer can be transferred, diluted, and processed directly to RT-qPCR. On the left, you see some results comparing this. At the top, you see pure RNA controls used to generate a standard curve. And then below, at the bottom, you see a spike in using an enveloped virus. The control here was a tri-reagent extraction and a standard curve determined through that. And then the same samples processed with the Q2A reagents. If you look at to the right, the CQ values versus the expected CQ values based on the standard curves, you see we have equivalent results, meaning no loss of signal. I'll touch now on some of the usability studies that we have done over the years. There is a significant interest in doing home collections using saliva. It is a non-invasive sample type. And DNA Genotek has taken these devices through robust testing over the years. Usability testing has been previously submitted to the FDA in support of our devices, the OGD 500 and the OGD 610. I'm sharing here with you part of one of such study to illustrate the robustness of that testing. We have evaluated both the comprehension of our instructions and compliance with collecting a reliable sample. These results support the use of the device for self-collection. We can leverage these results to support our customers. On the left is an example of comprehension of the instructions for use, and on the right we can see compliance to the instructions broken down by demographics. We see here the robustness of adhering to the instructions, which illustrates how easy the devices are to use by broad diversity of individuals. So to summarize, how DNA Genotek is supporting the COVID cases and EUAs, we are supporting all our customers and working closely with extraction and assay vendors. We are leveraging previous experience in collection and stabilization. We are generating technical data to support the use of DNA Genotek chemistries. And to date, several EUAs have already been granted using DNA Genotek devices. And now I'd like to turn over the rest of the talk to Dr. Wildemore.
Hi, everyone, and thank you for the opportunity to speak with you today. Once again, I am Dr. Bernadette Wildemore, the Chief Medical Officer from P23 Labs. If you're not familiar with us, P23 Labs is a new minority-owned molecular clinical lab in Little Rock, Arkansas, that is committed to creating accessible and actionable lab tests for all people. So conventional techniques for collecting respiratory samples for COVID-19 include some of the usual suspects, such as nasopharyngeal swabs or NP swabs, where a swab is inserted into the nose and guided into the pharynx or throat. Now, I've never had an NP swab performed personally, but reports are pretty consistent across the board. They're terrible. They're very uncomfortable. And in fact, a colleague who had an NP swab performed recently reported that she never, ever wants to be tested again. So as you can imagine, this is quite an issue, especially when you consider that there are populations that may need frequent and even repeated testing, such as healthcare workers. So at P23, we wanted to see if we could come up with something better. The assay we're offering is a real-time reverse transcriptase PCR test for a qualitative detection of COVID-19 DNA in samples collected by conventional methods. And we compare these with our new novel non-invasive saliva collection technique. A couple of things to point out here are, number one, is that this is a qualitative test. So basically, this will tell you yes or no, you have the infection. Second, our FDA EUA is currently limited to samples collected under the supervision of a healthcare professional. I can give you a little bit of secret information here if you promise not to tell, but we're currently seeking an EUA for at-home collection and hope to have this very soon. So P23 Labs has validated the performance of our SARS-CoV-2 assay that's including but not limited to LOD limits. Of course, we repeated the validation to bridge the results with saliva in the OM505 samples. Viral spikes and contrived or faked samples were used to demonstrate performance of the assay from the extraction through to detection. Of course, we wanted to focus on samples that were on the lower end of LOD. If you look to the right of this slide, I'm happy to report that if you compare the results of the negative control samples, these results matched what were expected in that the virus was undetected or could not be seen when there was no virus present. So saliva samples were in agreement with nasopharyngeal samples. Next, in this part of the study, we wanted to determine if the specimen was stable at different temperatures, specifically for shipping and storage purposes. And as you can see from our data, you can see that the samples did remain viable at room temperature and at extremes of the thermometer. Most importantly, this supports us for home collection and shipping completed at room temperature. Here we wanted to compare and confirm the clinical performance of samples obtained via NP swabs and our own saliva samples. Across all targets we assayed, we were able to demonstrate 100% concordance between positive and negative nasopharyngeal as well as positive and negative saliva samples when studied in parallel. Ultimately, we concluded that after our validation, we were confident that saliva samples could be used interchangeably and were a viable and much more comfortable option when compared to nasopharyngeal collection. So that wraps up our validation, and now I'd like to pass it back to Elizabeth. Thank you very much.
Thanks, Dr. Wildemar, and thanks to all of our speakers for the great presentation. We will now move on to our question and answer segment. Just before jumping into that, I would like to remind you that you can submit your questions in the chat box, and also that you will, or that all attendees will receive a copy of the recording following the close of the webinar. And with that, I'm just going to switch over to our questions. So the first question going to Dr. Wildemar, and you may have already touched on this, but the question is, what's the LOD and specificity of the test?
Okay, so the LOD and specificity of the test, I know that our metrics confirm that both the sensitivity and specificity of this test are well above 95%. Hopefully, this answers the question. If there continues to be questions, the specifics are listed online at our website, P23Labs.com.
Great. And another question for you, Dr. Wildemar. How difficult was it to validate saliva with the FDA?
So it was difficult, but fortunately they guide you through each and every step. The FDA challenges us to all meet a high level of excellence. So really, even though, yes, it was tough and sometimes frustrating, overall it was a worthwhile experience and hopefully allows us to provide the most accurate and reliable test to the customer.
Lovely. And for Mr. Rabuka, have there been any cases where at-home sampling collection is too complex for the average person to execute?
Well, for the most part, no. Through some of the early 510K clearance projects that we did initially with Origin DX and OraCollect DX, there are pretty stringent requirements on making sure that the instructions were easy to follow. We provided those studies to different population groups and different age groups as well, to be able to just to answer that specific question. It's really important to know that when you send a collection kit out like this, and and the instructions need to be followed properly, that they will in fact be. And there's aspects of our kit that we ensure that can be easy to use. So if you think about screwing the cap back on the tube after you collect a sample, well, to what torque does that need to be done, and do people with arthritis in their hands, can they handle that? So we've thought through those, and for the most part, we're in a really good situation that way, and have some data to show with that as well. Thank you.
Probably a question for Dr. Wildemar. Does the time of sample collection influence sensitivity of the test?
From everything that we have seen through our data from our validation studies, this really does not make a difference, the time of collection, no.
Okay, great. And another one for you. Can you describe how you compared the nasopharyngeal swab to saliva in the patients? Were the patients swabbed, and did they spit at the same time? And were they all symptomatic?
Yes. So these were patients that were clinically symptomatic. So they would have qualified, obviously, for testing. Now, as far as the first part of your question, we did our best to randomize the specimen collection to make sure that we had a good sort of cross-section of patients to be able to have the answers that we reported.
Okay, great. I think this is a similar question, but just in case you have anything further to add, is a saliva sample collected first thing in the morning any different from a random collection?
Again, going back to my previous question, based on our data, it doesn't matter. It's not like you have to wake up and then, spit in the tube. It really can be collected at any time during the day.
Excellent. Question, I'd like to know more about P23 Labs demographics. You mentioned it being minority owned. What is your perspective on the prevalence of gender and ethnic racial diversity in STEM and in biotechnology?
Woo!
And I can repeat that.
Yeah, I can see the question here. That's quite a question. I'm very proud to work for P23. The founder of our company is an African-American female. And obviously, I am a female as well. With the two of us being involved with the company, we put a high priority on finding other people who share our vision for the company. And so, obviously, we try to pay attention to finding underrepresented minority groups to join our fantastic team. Any more about this? It's a little bit longer of an answer, so I would be happy to follow up with the question asker at a later date.
Sounds good. And did P23 Labs contact the FDA for guidance on validation prior to submitting an EUA request?
We did, yes.
Great. And what is the correlation between stage of disease and sensitivity of the assay?
Is this another question for me?
It is, I believe.
And I'm sorry, can you repeat that question, please?
Sure, of course. What is the correlation between stage of disease and sensitivity of the assay?
Oh, I'm sorry. I was having a little bit of a technical issue there.
That's okay.
Again, we used randomized patients for our validation, and so we really have not performed that study, meaning, to sort of confirm the clinical stage of the disease with our ultimate results. But so far, we have not seen any differences among the patients that we've tested so far.
Okay, great. Sorry to hit you with another question. Can you describe the shipping protocol for at-home collection COVID-19 tests? And are shipping protocols for COVID-19 tests different for other Origin home collections? So maybe the first part of the question for you, and then maybe Scott can speak to the second.
Okay, so As far as shipping, now as I've mentioned, the EUA that has been granted for us does not cover independent home collection just yet, meaning that a person cannot by themselves submit the sample. However, if the sample is collected under the supervision of a healthcare provider, the patient can do this process at room temperature, as I mentioned during my part of the talk. In our kit, we have mailing information where the patient just basically pops the tube that they have spit into this kit, and then they mail it back to our lab in Little Rock, Arkansas. It's actually very simple.
Yeah, I'll add just a couple quick comments. We've looked at the UN 3373 guideline for shipping infectious samples, or known to be infectious samples. At the end of the day, I'll speak more, in the pre-COVID world, the onus is on the shipper to declare a package as being infectious or not infectious. Obviously, when you're a layperson collecting at home, you may not realize you're infectious or you have some kind of infection. So typical packaging for at home would comply with the IATA rules for shipping infectious samples, which include biohazard bags, secondary packaging, and then our primary package. So the tube has been tested with multiple freeze-thaw cycles, various vacuum pressures to ensure its safety in all situations from the point of collection to the lab. So there's a lot of thought that's been put into that, and then more recently, as Rafal shared, the inactivation studies that we performed to ensure that the personnel handling the sample in the lab will have as much minimization of the risk happening, but before the sample gets to the lab.
Excellent. Okay, so the next question, perhaps a mixture between Scott and Dr. Iwasiow. How do I get sample collection kits for evaluation for Kenya? This question is coming from Dr. Ida. I hope I have pronounced your name properly. And the second question from him is, can you help in implementation of the RT-PCR for qualitative detection of COVID-19 using saliva in Kenya?
So I'll take the first. This is Scott. With regards to getting samples within Kenya to try, we have a distributor that we can connect you with, and one of our account managers will follow up with that after this meeting. So thank you for that question. Rafal, I'll let you speak to the PCR implementation.
Sure. So as far as the assay goes, that really goes back to the manufacturer of the assay. But at DNA Genotek, we're here to support the use of our kits. We can certainly provide guidance based on our experience around evaluating the kits in those assays. So encourage you to reach out to our tech support team.
And I will be sharing the tech support email address later on, so just stay tuned for that. The next question, Dr. Iwasiow, I think this goes to you again. Is there a chemistry difference or other difference between the Oragene DX and OmniGene oral kits? And are both kits considered to be an option for diagnostic testing?
Yes, the chemistries are different between those kits. There are, in fact, EUAs now with both of those kits in the field. So they do have a different modes of performance. Our primary positioning is the OmniGene oral kit, which was designed for microbial RNA and DNA kits. The Oragene DX have gone through extensive FDA clearance for human genomics testing.
Great. Good. And another question for Dr. Wildemore, do you experience better results with the swab versus saliva?
So that's a terrific question. Thank you very much. As you can see from the data that I've presented, there are similar results. However, in fact, saliva actually might be better in some cases because there's less variability in the techniques for collecting this sample. Ultimately, I think the benefits of saliva over a nasopharyngeal swab are obviously, much more comfortable for the patient. And next, it is much less likely to create an infection issue for the collector, meaning that there's not as much risk for the person trying to collect the sample. So really, I think they're quite similar, and the data supports that.
Good. And Dr. Wildemore, another question for you. How can I get a package insert for your assay?
Well, thank you very much for your interest in P23 Labs. Absolutely. You can visit our website, P23Labs.com, and go there and contact us via email, and we will try to get the information out to you.
Good. And another one for you, are there specific temperature requirements for shipping the sample?
Another good question. As I was mentioning during my talk, we were able to show that shipping the samples at room temperature was completely acceptable according to our results. And you may also have seen that we tested at what I like to call the extremes of the thermometer, so both very, very cold as well as very, very warm or hot. So now that we're going into the summer in the United States, we definitely want to be sure that any samples that are sent to us in Little Rock, Arkansas, are stable and valid, as they are coming to us really in the south of the United States. And so, fortunately, we have data that supports the validity of this type of sample. Thank you so much for your question.
No problem. Okay, and I think, unless I have missed anything, I believe that is our final question. So with that, I will, conclude today's webinar. Thank you, Mr. Rabuka, Dr. Iwasiow, and Dr. Wildemore for your expertise, and thanks to all of you for joining us. If you'd like to learn more or speak with us about how DNA Genotek's products might help you overcome sample collection challenges, please contact us at info@dnagenotech.com. That same email address can be used for any technical related questions. Alternatively, for any product or technical questions, you can reach out to Mr. Rabuka or Dr. Iwasiow directly. If you'd like to request free trial kits, you can do so at try.dnagenotech.com/free kits. Any questions for P23 Labs can be directed to info@p23labs.com or to Dr. Wildemore directly. Her email is on your screen here. And for information about P23 Labs or to order the COVID-19 test, you can visit their website, and that's p23labs.com. I thank you again for spending the hour with us. Please enjoy the remainder of your day.